Imagine if you could fix a spelling mistake in a recipe just by using a special eraser and pen. Now, think even bigger. What if scientists could do something very similar but not with words, with the instructions inside living things? These special instructions are called genes, and they're like recipes hidden inside every plant, animal, and even you. Genes are made up of DNA, which tells our bodies how to grow, what color our eyes will be, or even how tall we might become. Sometimes, there are little mistakes in these instructions. Scientists wondered for many years if it was possible to correct these mistakes. But changing something so tiny and important sounded impossible until they invented tools for genomic editing. Genomic editing is the way scientists can change, remove, or add parts to the instructions inside a living thing's cells. One of the most famous tools is called CRISPR, which sounds a bit like a superhero. Let's imagine your DNA as a really, really long train made of colorful blocks, where each block is a letter in the instruction. If there's a block that shouldn't be there, CRISPR is like a super smart pair of scissors that can zoom in, snip out the wrong block, and help put the right one back in place. CRISPR works with a tiny guide, a bit like a treasure map, that shows the scissors exactly where to look on the DNA train. Once CRISPR finds a spot, it cuts the DNA, and then the cell does one of two things. Either it glues the ends back together, sometimes losing a bit of the message, or it uses help from scientists to add a new piece of correct DNA. Long before CRISPR, there were genomic editing tools called zinc finger nucleases and talons. Think of these like older, less speedy versions of CRISPR. They worked like careful locksmiths, but they were much slower and harder to guide. With these special editing tools, scientists have fixed genetic mistakes that cause sickness, made plants that can resist bugs or droughts, and even studied how different genes work in animals like mice or zebrafish. But like all powerful tools, genomic editing must be used with care. There are many rules and debates about what's safe or fair when changing genes. Scientists discuss these questions often, making sure they only use genomic editing to help people, animals, and the planet in wise ways. So maybe one day, thanks to clever tools like CRISPR, we might be able to heal more diseases or even grow better food just by correcting the recipes written deep inside every living thing.